Focus everyone, because now it's time for bacterial conjugation, which is of course another type of horizontal gene transfer. As we have studied before in the introductory lecture of bacterial transformation, that horizontal gene transfer are of three types: transformation, conjugation, and transduction. We have already studied transformation, so now it's time for conjugation. In transformation, the bacteria used to get the genetic material from a dead bacteria. The genetic material in this case used to be present in the extracellular fluid, floating around and just come by the bacteria by chance. But in case of conjugation, two bacteria participate in a very specialized process by forming a bridge between those two cells. So, conjugation is the type of horizontal gene transfer where genetic material passes from one bacterial cell to another by direct cell-to-cell contact. The bacteria that provides the genetic material is called donor bacteria, whereas the bacteria that receives the genetic material is called recipient bacteria. So, what differentiates these two bacteria? It is the presence of a very specific site in the genetic material of the donor bacteria called origin of transfer, or ORIT site, or sometimes simply called origin or O site. Another factor that defines a donor bacteria is the presence of some transfer genes, which together form an operon called operon. So what is this origin of transfer or O site? Both plasmid and chromosomal DNA in the bacterial cell are double-stranded. But during conjugation, the DNA that gets transferred between the two bacterial cells is a single-stranded DNA. That means at some point the bacteria has to cleave one of its strands in one of its genetic material, either plasmid or chromosomal DNA, and transfer it to the recipient cell. So for example, if this is a plasmid, it is of course a double-stranded DNA, then at some point the bacteria will cleave one strand of the genetic material and pass it to the recipient cell. The site where the bacterial cell nick its DNA is called the origin of transfer site. Why origin of transfer? Because it is this point where the transfer begins. This cutting of gene is an enzymatic process. For example, one of the enzyme known as relaxase is involved in cutting the plasmid and producing the single-stranded DNA. Another property of the donor cell is the presence of operon or transfer genes. These transfer genes together produce various proteins and enzymes that participate in the process of conjugation. So the transfer genes produces proteins known as transfer proteins. Pretty simple, huh? One group of protein form a structure known as transferosome. Some protein form another structure known as relaxosome. And remember the relaxase enzyme I just talked about? Relaxase enzyme is a part of this relaxosome complex. Some other transfer proteins together forms the pore. That is this part, this narrow bridge or pore that is formed between these two cells. This bridge is also known as conjugal bridge or mating bridge or conjugal tube or mating tube. You should remember all these terms because the question can come with any of these terms. During the formation of conjugal bridge or mating bridge, the donor bacteria first forms a protein structure known as pilus, or in plural pili. Remember that pilus and pili has a single L. When this pilus comes in contact with a recipient bacterial cell, it gets attached with the bacterial cell and thus form the conjugal bridge. Of course, it has a lumen within it through which the genetic material can pass from the donor cell to the recipient cell. Now listen very carefully. Depending on where this origin of transfer site and transfer genes are present, the donor bacteria can be divided into three types. These three types are A+, F', and HFR. So what is A+? When the origin of transfer site as well as transfer genes are located in the plasmid of the bacteria, then the donor bacterial cell is known as A+ cell. Remember that these plasmids that contain the origin of transfer site and transfer genes mostly contain genes related to the conjugation process and no chromosomal gene. If somehow the plasmid containing the origin of transfer and transfer gene contains some chromosomal genes, then it will be known as F'. So remember that in both F+ as well as F', the origin of transfer site and the transfer genes are present in the plasmid and not in the bacterial chromosome. The only difference is the presence of chromosomal gene or absence of it within that particular plasmid. In the third case, the bacteria contains both these origin of transfer site as well as transfer genes within the bacterial chromosome and not within some other plasmid. So in this case, the process of conjugation are regulated by elements within the bacterial chromosome. The recipient bacterial cell, on the other hand, does not have any origin of transfer site or transfer genes. These cells are always named F- cell. So when F+ cell transfer its genetic material to the F- cell, this type of conjugation is known as F+ F- conjugation. When F' cell does so, it is known as F' F- conjugation, and when an HFR cell does this type of conjugation, it is known as HFR F- conjugation. So these three, A+, F-, F', F-, and HFR F- are the major types of conjugations, and I shall discuss them one by one, starting with A+, F- conjugation in the next video. If you found this video useful, please like it. Also don't forget to subscribe and share it with your friends. See you in the next video. Thank you for watching.